beautiful Friday here in Oklahoma. The sun is out, the wonderful and beautiful sun, or the energy giving and life sustaining sun is out and shining in a beautiful way this morning. So, we're here with another Chip Talks to talk to you about something equally exciting. It's something, and I'm being a little bit facetious when I say exciting. I'm sure you all are glued to your chairs and waiting to hear about microRNA. This is sort of a necessary talk that we need to go through to get to some sort of conclusions that I want to get you guys to. But it's important to understand microRNA and its role in the human body and kind of what it does, because it's not that you've been mistaught, you've just kind of been, let's say, misled a little bit. So DNA, so we have this understanding and impression of kind of what DNA is and how DNA works, don't we? And so, what is that? Well, we've seen the strands, and somehow that stuff happens, and stuff gets translated from that, and it causes changes in our body, right, that are genetics. It's kind of how I thought about it in the past. It's just like DNA. I get it. It codes for genes and genetics, and it looks like that, and RNAs involved kind of, and there's stuff that happens inside of a cell, right? Well, sorry, we got to dive a little bit more into detail of everything than that, but that was my understanding. And again, that's kind of, I think, most people's rough understanding of what happens. Well, wow, is it way more interesting and kind of cooler than that? So we'll walk through that here real quick like. So we're going to talk about, obviously, microRNA today, but we need to have a basis, a starting point. So let's talk about DNA, first. So, and this is stuff, by the way, that happens inside of a cell. And, I'm sure you guys were equally as bored and frustrated with this, when I learned about it in high school or whenever I learned about it, and it was sort of a memorization exercise rather than a learning experience. But this is kind of cool stuff. So anyway, just bear with me for a little bit, because this is all going to, believe it or not, tie back to diet and behavior and all that stuff, right? Because that's how we affect all this stuff. But we need to understand how we work in a little bit better way than DNA, RNA, genes, right? So what really happens here? And, how dynamic is this? How static is this? Like, if you're like me, I mean, somebody tells me, here's your genetic profile. To me, that feels like it never changes. If things are off, does that mean I'm screwed forever? That's kind of the way that it feels. And gosh, is that the furthest thing from the truth? So all of this is extraordinarily dynamic. To say that, oh, I got my genetic profile done. Ah, good. That changed 30 seconds later. Good for you. It really, it's interesting information, right? And there are things that potentially are on or off that, are important to note, but it's dynamic. Let's just put it that way. 43% of your genes will change just throughout the day by the motion of the sun. No joke. And literally change according to circadian rhythms that are run by the sun. So how does all this work? Well, we should understand it a little bit better than perhaps we did in high school. And so let me see if I can walk you through this in a little bit more interesting and entertaining way than Mr. Smith did in high school biology or wherever we have this stuff. So let's start with kind of DNA. So what is DNA? What makes up DNA? That's pretty important. Well, what makes up DNA are things called nucleotides. And again, I'm always looking for sort of the lowest, expression of stuff, highest truth, I guess is the way to say it. But, what's the most basic stuff inside of a cell, right? Granules, and so those become things, which is interesting with electrical stimulation, but we'll talk about that down the road. So that's way down the road. Right now, let's talk about nucleotides. So nucleotides are these five guys. So what are the five guys? Thymine, adenine, cytosine, guanine. Well, I guess it's, four guys, but TACG, right? So those are the guys, and they're what they look like is over here on the left. So that is what a nucleotide looks like. But these are the guys that make up DNA. So DNA is just made up of these different guys, and that's how DNA works, is it's made up of nucleotides, and nucleotides go together in the strand, right? The strand that we're used to seeing. Okay, so what happens from there? Did I spell messenger wrong? I may have. Sorry about that if I did. What happened? Spelling is not my thing. This is my thing, but spelling isn't. So what happens from there, right? Well, if you look at over on, well, the best way to, I guess, look at this is on the graph on the left. So what the blue stuff is there is DNA, and that's DNA that's in the nucleus of a cell. What is that DNA? Well, that DNA has every single gene, that you need on that DNA. So it's a big DNA strand, and depending on where you are on that DNA, that will be certain genes. Well, how do genes express? Well, again, the messenger RNA has to come along. It's got to basically, through this thing called RNA polymerase, it's got to find that section on that DNA strand, and then it copies it into messenger RNA. There's some sub-processes there that we're going to talk about, because before it becomes messenger RNA, a lot of things can happen to it, but basically that's, when we talk about the vaccine, the COVID vaccine, we say, yeah, we're using messenger RNA. What? That's, golly, that's stuff you want your body to make. You don't want to inject inside of you. And again, there's ways to, change kind of how your body's making this, but you don't want messenger RNA injected in your body. That can cause massive changes inside of you in the way that this is. So it can cause genes to express, de-express, all that. What is that messenger RNA that was in the vaccine? That would be interesting to find out, wouldn't it? So anyway, but messenger RNA is really how you do amino acids, okay? And so if you look at kind of what the function of a cell is, well, cells make protein, so we all kind of were taught that, right? Well, what does that look like? Well, again, we have to have DNA. DNA is what starts the process. You have an RNA polymerase, which is going to read that DNA and then make messenger RNA from that. And then that messenger RNA basically is used to build amino acids. So this is the way that all amino acids are built. And you can see from this table how, I guess it's 16 amino acids are built, or maybe even more than that. But how a lot of the amino acids are built, but all amino acids are just built from nucleotides that come from messenger RNA. Okay, so are you guys with me so far? It's really, this isn't too horrible and complicated. It's just a little bit different than what you were taught in high school. So again, DNA is in the nucleus of a cell. RNA polymerase will break him apart. Messenger RNA will sort of build from a little piece of him. That will go out and become part of an amino acid chain, which will become a protein. Because amino acids are how we build proteins, all right? And again, you can back this up into what is a protein. Well, it's just amino acids, which are just nucleotides. So when you're eating protein, it's important to know what those amino acids are that make up that protein, all right? Because there's lots of different kinds of protein. I mean, you signal with I'll just read you this. Amino acids are the building blocks of proteins. Proteins are long chains of amino acids. Your body has thousands of different proteins that each have important jobs. By the way, when you eat meat, and you're eating that protein in meat, you're eating exactly that. The body of that cow or whatever has thousands of different proteins that each have important jobs. So again, you're eating it in muscle probably, so it had an important job in muscle, but you don't know what's in that. You don't know what amino acids are in that protein. You're going to go by what the USDA tells you on a general graph. That's probably a bad idea, for your health. And this is why we recommend supplementing with amino acids. Each protein has its own sequence of amino acids, absolutely. Again, if that cell doesn't have the amino acids it needs to build that protein, what happens? It doesn't. It won't build that protein, and you will suffer in some way. Again, the cell's trying to build a protein to do something, to help you in some way. Everything in you is always striving toward perfect function. You're always striving to get healthy, always striving to get better, always striving to defend yourself. You need proteins to do that, okay? Important stuff. Proteins run you. So again, proteins are responsible for nearly every task of cellular life, including cell shape, inner organization, product manufacture, and waste cleanup, and routine maintenance. Proteins also receive signals from outside the cell and mobilize intracellular responses. How do they do that? That's where we're going. Light, baby. But anyway, so proteins also receive signals, and again, there's a mechanism for doing that, that's down the road, but that's where we're going. But proteins also receive signals from outside the cell and mobilize intercellular responses. So again, these proteins are sensitive, they understand changes in environment, they signal back to cells, but these proteins are a super important part of cellular function and kind of what you're trying to do, how you work and how you function. Proteins are super important, okay? All right, so DNA, RNA, the little m RNA means messenger RNA. So again, that's important. That's like genetics. That's like genetics, really. That is genetics. So messenger RNA is genetics and how genes express. So, you could think of that. The way I used to think of DNA, I guess, is how I now think of messenger RNA. And then there's microRNA. MicroRNA can silence messenger RNA or DNA or RNA. So microRNAs are super important to understand because they're little guys that are inside the cells and they can shut down or enhance any messenger RNA process, which means any genetic process, okay? So hopefully you guys are seeing how dynamic this is. I mean, this is just all the time. All this stuff is changing, and we think of ourselves as, oh, I got my 23andMe genetic thing, and this is me. No, it's not. Not even close. All right, so microRNA, what is microRNA? Well, again, it sort of starts through the same processes. It's, going to come off of the same, it actually is read by a different RNA polymerase than messenger RNA, which is interesting and, let's say probably important that it's a different RNA polymerase. But microRNA sort of comes off in the same way, so it's read off of a DNA strand and builds just these little short segments. They're kind of like messenger RNA, but kind of not. They get stuck, and they're double-stranded, so microRNA are double-stranded. They can get stuck, they get cut up and spliced inside of the nucleus of the cell, then some of them stay in there and again modify things inside of there. Some of them get exported to the cytoplasm of the cell and they do things there. And again, what do? Well, they interfere with our whole DNA process and they get in the middle of our DNA process. Where do they come from? That's an interesting story. But anyway, this is kind of what they do, and they look to be our friends in certain ways, but they also don't look to be our friends in other ways. So here's what looks like kind of what happens, not looks like, this is exactly what happens inside of you as to how you work, so again, you have a fuel side of you, which is, glucose, fats, or light, and that's the fuel side of kind of a cell and how a cell generates fuel and energy. You also have, what's a cell trying to do? What's its function? What's its purpose? Which leads to kind of what you're trying to do, right? And sort of survive is the biggest thing. But, how do your cells all go together to sort of make that symphony? Well, it's important to understand how a cell works and what a cell is trying to do. So a cell is trying to generally make a protein. That is the purpose. It's a little, he sets out, and what is your purpose, little cell? To make a protein, okay? So, and so how does all this work? So first of all, DNA is transcribed by that's actually RNA polymerase to pre-RNA, okay? So again, inside of the nucleus of a cell, you got this giant thing that's DNA that has every single gene in you coded, okay? When you need to transcribe a gene, when your body says, oh, well, it's time I need to transcribe a CB1 neuroreceptor to be able to work your endocannabinoid system. So again, cells got to go to RNA polymerase. That's got to go find that place for CB1 on the DNA. That'll make a little messenger RNA string, and off it goes. That can also make a little MicroRNA string, and that might be just a part of CB1 and might silence that messenger RNA. Okay, so several things can happen there when sort of that DNA comes apart. But then after that, both the messenger RNA and the microRNA have to sort of be spliced and cut up, and they go through the endoplasmic reticulum, which is a process. And then in certain times, they get sort of popped out of the nucleus of the cell to go do work. Well, microRNA can, and so I'm now in the middle bullet here, but microRNA can silence or promote messenger RNA by overlaying important coding segments. So microRNA can alter messenger RNA, and messenger RNA, again, think of that as what we think of as DNA, I guess. It's out there trying to do work, okay? So, but microRNA can overlay. And again, what does this do? Well, protein expression and thereby function can be altered by a cell at any time by microRNA. Again, so if I can overlay your DNA, I guess is the easiest way to think about it, but that's actually messenger RNA. So if I can overlay that and change the expression of that, again, I can change the expression of protein, and I can change what a cell is trying to do. I can shut it down, I can make it different, I can change it. And this looks to be exactly what bacteria, viruses, funguses, this thing called the bacterial hijack does, and how bacteria are able to take such tight control of our immune processes in certain ways. Okay, so does this show up in things that we think are bacterially related, like cancer? Yes, and like crazy. So again, in the classical microRNA synthesis pathway, mature microRNAs bind to the 3' UTR, their target messenger RNA, through complementary base pairing, which leads to the degradation of target messenger RNA or inhibition of protein translation. So in a way that we just described, again, in cancer, this happens with microRNAs in certain instances, binding to messenger RNAs and shutting them down or changing their function. So this graph here, the A and the B, so let me just kind of read you through this, because I think some of this is pretty technical and it's not important that you remember metal 3 or FTO or any of these genes. It's just important that you remember that this happens and that really microRNA causes this to happen. And M6A is a really important player in this, but M6A modifications play different roles in multiple cancers by influencing the maturation of microRNAs. M6A modification promotes microRNA maturization by recruiting DGCR8 to promote microRNA processing. That means inside of the nucleus of the cell, or increasing microRNA splicing by dicer in different cancers. So that gets upregulated in certain cancers, that activity that's in the nucleus of a cell. Furthermore, M6A modification can promote or inhibit the occurrence and development of tumors by affecting the maturation of microRNAs. Now, that's a pretty important statement. M6A modification can promote or inhibit the occurrence and development of tumors. What? Gee, with billions of dollars of cancer research, I think I'd be going at that little fella, don't you guys? M6A modification can promote or inhibit the occurrence and development of tumors. How? By affecting the maturation of microRNAs. It doesn't get any more basic than that, boys and girls. That's right off your DNA and sort of how things appear to be different in cancer versus normal and M6A. Okay? Important. that big long string of letters, HNRNPA2B1, has also been found to inhibit the shearing of some of the pre-microRNAs. However, the specific mechanism underlying this inhibition remains unclear in breast cancer. In this B side, it says microRNAs affect cancer progression by regulating M6A modification of messenger RNAs. Let me read that again. MicroRNAs affect cancer progression. So we've got inhibition or promotion of tumors, and we've also got progression happening around this M6 thing and the genes associated with it. I'd say that's pretty important, wouldn't you? And around microRNA. So microRNA, basically, point of all this slide, microRNA influences M6A. M6A has to do with how quick tumors grow, or if they grow at all, and inhibiting them or making them grow faster. Pretty important stuff. Very important stuff. Okay. What else? Does this have to do with other diseases? Yes. It turns out in autism, another disease that I've very tightly studied along this sort of bacterial hijack, it turns out this also happens. So listen to this. MicroRNAs, small non-coding RNAs that have been recognized as critical regulators of gene expression, play a key role in the neurodevelopmental transcriptional networks of the human brain. Previous investigations have proven that circulating microRNAs open up new possibilities for the emerging roles of diagnostic and prognostic biomarkers in human disorders and diseases. So again, if you got microRNAs showing up, and if they're of a certain type, then that's a pretty good indication that you might be going down a disease path. And again, why is this? Because bacteria make these guys and they show up in your circulation. They show up in saliva quite readily, which is kind of cool, which means, spit tests are cool. Due to the complexity of autism spectrum disorder, considerable endeavors have either unsuccessfully identified biomarkers for the disorder or have not yet been established. If you live with autism spectrum disorder, again, I'm sorry, first of all, for the state of care, for the state of pharma, for the state of, research, for all that. It's just a pathetic freaking mess. You can't even get diagnosed until you're 6 or 7 years old. How do you get diagnosed? They put your child through a series of tests, and they'll grade, them by these tests. So there are no markers. I can't give you a test right now, a saliva test to see if you have autism. But, it turns out if you have autism, you have these microRNAs, and they are prognostic. And they are good biomarkers to indicate autism. That's pretty damn important, boys and girls. That's pretty important. The other cool thing is, again, you can get these right off of, PCR, right? So there's microRNA. You can test for all this, I guess, on a PCR. Cell-free circulating microRNAs and biofluids are extraordinarily stable and considered to represent the next generation of clinical non-invasive biomarkers for many pathologies, including neurological and neurodevelopmental disorders. Yeah. So where is testing going? Right here. All right. MicroRNA, and again, right here on this graph, which we can blow up if we need to. It's kind of hard to read. I didn't cite this study. If you want to know this study, ping me. But you can kind of see, let's blow this up just real quick. Just because you can see that, and in saliva, that's kind of what I'm interested in. I don't want to stick a needle in you, right? So you can see in these studies, over here on the right are the studies, that you can see kind of what microRNAs were downregulated and what microRNAs were upregulated. And this is super interesting as, you begin to kind of, if you want to, begin to study these microRNAs, you're gonna find a lot of commonalities with things, which is interesting because these are the guys that are sort of mutating in cancer, in autism, in most severe disease. You have certain mutations, gene mutations that always take place, like P53. Well, what mutates P53? MicroRNA, okay? You seeing it yet? Hopefully you are. MicroRNA and other disease. All right, so it's, what about other disease? Well, microRNAs have emerged as promising candidates with extensive diagnostic potential. And again, we're talking about testing, we're talking about IDing things, we're talking about pharma targets, we're talking about all that. MicroRNAs. And why? Well, due to their role in numerous diseases. And again, they're part of the bacterial hijack. So any disease, and not all disease, I say it all the time, all disease is bacterial. That's wrong. And again, I can think of 6 right off the top of my head that aren't. But a lot of disease are, okay? And so in the diseases that are, what's the tell? MicroRNA. And it's measurable. And it's measurable with a PCR in a 20-minute test. So microRNA biomarkers offer benefits as to the diagnosis, management, and treatment of infectious disease. What? MicroRNA biomarkers offer benefits as to the diagnosis, management, and treatment of infectious disease. Why? Because I don't need a mass spec. I just need a PCR. In fact, some of these you can do with dye and stain tests, right? So they're really easy to do. Investigation of these novel biomarkers could advance the use of personalized medicine. What's that mean? Oh, I can diagnose myself? Personalized medicine? In infectious disease treatment, which raises important considerations for validating their use as diagnostic or prognostic markers. And clearly they will be and are. And again, this is right where I'm going. I'm sure other people are as well. Finally, we discussed new and emerging microRNA detection platforms with a focus on rapid point-of-care testing to evaluate the benefits and obstacles of microRNA biomarkers for infectious disease. Okay, hopefully you're seeing how important this is. It's pretty dang important, right? It's really important. So microRNAs are a tell. Now, how this works, again, this is new, right? So this is new in research. Old Chip has to rely on other people. Like, I read other people's research. That's what I cite. That's what I go try to find when I'm trying to put together the pieces of the puzzle. I'm about out of my runway, so I'm about out of existing research. So, hopefully at some point out there, somebody will say, well, Old Chip might need a lab and some minions to go figure this stuff out. And that'll happen, at some point when God's ready for it too, or, however that'll work. Anyway, but until now, there is enough research to continue to educate you all on the wonderfulness of how you work. God built you a really cool vehicle. And that vehicle is closely associated with that bright, shiny ball in the sky. And, that's ultimately where we're headed and where I'm taking you. But I want you to understand more about how you work, about how we look at disease, about what disease is, how you've been misled about, things. So, that's what we're all about here. So anyway, you guys have a good weekend. Thank you very much, and love you guys.